If we remember that DNA has 4 types of bases, adenine, thymine, cytosine and guanine. 3 bases code for 1 amino acid. It is also non-overlapping. So this means that the base code is read in groups of 3. So for example, CAT will code for amino acid 1. Then the next 3 bases are read, so TCT for the next amino acid 2, and so on. The code is also degenerate, which means that there could be more than one triplet code that will code for the same amino acid. So for example, TCT or TCC or TCA or TCG all code for the amino acid serine. A mutation is a change in DNA. If the mutation is a substitution, one of the bases of the triplet code has been substituted with a different base. This would then change the triplet code, which in turn may affect what amino acid that triplet code codes for. For example, TCT codes for serine. If a substitution occurs that replaces the C in the middle with adenine instead, and mutation has caused that change, then the code is now TAT, and that will code for a different amino acid, tyrosine. If a substitution happens in the 3rd base of the triplet code, this would be known as a neutral mutation. So for example, if TCT, TCC, TCA or TCG, the 3rd base was changed, they all code for the same amino acid serine. And this then shows that there is no effect in that 3rd substitution, and that is because the code is degenerate. More than one triplet code for the same amino acid. A deletion mutation is the removal of one base, and this would cause what is known as a frame shift. So we're looking in this example where we have 4 sets of the same code, CAT, for the same amino acid, HIS or histidine. If the second A is deleted, then this will cause the frame shift to happen. The first 3 triplet code is not affected, but from there on it is, and that's why it's a frame shift. So the next triplet code becomes CTC, which codes for a completely different amino acid, as does the next. So we have shifted along one, which is changing the amino acids. And this would then change the structure of the protein being formed and could affect the function or its ability to do its job. So it is known as a frame shift because from the point where the deletion occurred, from that point onwards, the code has been affected. Chromosome mutations is when large sections of DNA are affected. Generally, it is when the homologous chromosomes, the chromosomes that are the same pair, do not divide or segregate properly during meiosis. Two examples to be familiar with: Down syndrome, and this is when during meiosis pair number 21 do not segregate properly. The second is Turner syndrome, and it is failure of the sex chromosomes to segregate properly, which would result in the child having only one sex chromosome, the X chromosome, being female but sterile.